This course is going to be an introduction to the course Biochemistry. And what is biochemistry? Biochemistry is a course that studies of various molecules that make up of living matters or organic matters. So living matters are made up of varieties of molecules like carbohydrates. Let's just list them out. We have the carbohydrates. The carbohydrates is one of the molecules that are present in living matter and their cells. So we have lipids, we have fat proteins. These lipids can rather be called fat and oil. We have vitamins and minerals. We have enzymes. Enzymes can be proteinous, and we also have enzymes in general. Then we have water. These are the compounds that comprises or that are present in living matters. They are the major components of living matters. Actually, water is about 70% of living matter, of living things. But anyway, we are going to start with carbohydrates today. Since biochemistry is a study of, some regard this as study of food molecules, while some also regard, because it is regarded as food molecules because most of these components of living matters are derived or are gotten from foods. But we are going to start with carbohydrates. Definition of a carbohydrate. Carbohydrates are compounds that contain carbon skeleton and aldehyde or ketone group. Aldehyde is CHO group, or rather you can write it as H, C, and so this is an aldehyde group, and this, or you call it alkanal or the anal group. Then we have the ketone. So a particular functional organic radical can be attached to this. So carbohydrates do contain carbon skeleton or carbon chain, which is attached to what? Either aldehyde group or ketone group. And this group with the carbon are surrounded by hydroxyl group. So this is the qualification to call a compound a carbohydrate. An example of the carbohydrates is glyceraldehyde. Let's look at glyceraldehyde. If I have C, H, OH, H, then with C, O, H. This is glyceraldehyde. So it's also a carbohydrate. It has the carbon chain connected to the what? Aldehyde group, and this particular carbon, they are what? They are connected to hydroxyl group. They are surrounded with hydroxyl group. Now, each carbon present in these carbohydrates are chiral carbon. What do I mean by chiral carbon? If I have a carbon C and I have another one, this one is connected to hydrogen, while this one is connected to hydrogen and OH. This one is regarded as a chiral carbon. Why? Because it is connected to atoms, two species of atom other than H. Why this one, the only type of atom present in it or attached to it is just what? One, hydrogen. Why this one is connected to two species of atoms or two. So it's called chiral carbon. So this chirality of carbons that are present in carbohydrates enables them to form isomers. So when we say isomers are compounds that would exhibit isomerism. So isomerism can be defined as the existence of compound with the same molecular formula but different structural formula. So example of such we have them, but I'll first of all group the isomerism or the type of isomers carbohydrates normally form into two, because we have the geometrical and we have the optical isomers. The geometrical isomers are just different in their arrangements. Because we have the cis and we have the trans. Let's take a look at malic acid. Malic acid. These geometrical isomers, arrangement of the functional group in a double bond carbohydrate like this, like in malic acid, RCOH here, and H here, H here, and COOH here. So this is malic acid. So as we can see, this type of arrangement is what we call the cis. So comparing it to another acid called fumaric acid, and that contains HOOC with HH. Fumaric acid. So these are what they are, geometrical isomers due to the arrangement. So this one is called a trans, because due to the arrangement of the what, they are in slant position to each other, while this one are what, parallel and straight line position to each other. So these are one type of isomers. Then we also have geometrical isomers. These are geometrical isomers. We have optical isomers. These are called enantiomers, or they are just like a mirror image of each other. Let's take a look at this compound. See, OH, and this compound. If we take a look at the differences is just this particular point where the configuration of the OH, the arrangement of the OH is different from one another. It's just that the configuration of the OH around the chiral carbon is different. So one is here and the other is here. They are just like a mirror image of each other. So these are what enantiomers or optical isomers. So this is one of the type of isomerism carbohydrates do showcase. Now we also have that's on properties of carbohydrates and introduction to carbohydrates. Let's quickly look at epimers. There are some compounds in carbohydrates, we call them epimers. Epimers are compounds that has the same, they actually have the same molecular formula, but the difference is the arrangement in their, just similar to this, similar to something like this. The difference is just like they are formed as a result of optical isomerism. Let's look at glucose as an example. Glucose, D-glucose. This is a diagram to represent the molecule of D-glucose. This is D-glucose. It is called D-glucose because of the configuration of OH, the rotational, the spatial arrangement of the OH, So this D means dextrorotary glucose, meaning that what the OH is rotated towards the right. So that's dextrorotary glucose. Now let's compare it to another epimers like manose. So manose is an epimer of glucose. So the difference in it is just this is 1 carbon, 2, 3, 4, 5, 6. At the number 2 carbon, at the second carbon of the manose compared to the glucose, there is a change in structure, just like what takes place here. So we have the HO here and the H here. So we have OH, H, OH, which is the hydroxyl group. So the only difference is that this second carbon, this second chiral carbon present here. So this is called D-manose. This is called D-manose. Now let's look at another compound called galactose, which is also an epimer of glucose. Galactose is also an epimer of glucose, and the difference in arrangement of galactose is just at the number 4th carbon. So if you look at it here, the difference is clear. So at the number 4th carbon of galactose, the same structure will be present in galactose. So let's just give it a drawing. We have H, so here, if you look at this, we are going to have H here and OH here. So here we have H, so OH, or HO, supposed to be HO. And here we are going to have just what is here, OH and H. So this is the galactose. So these are the epimers of glucose. They are the epimers of glucose due to the difference in arrangement of those position of carbon. Moving on, proceeding on the epimers, we are going to look at the classes of carbohydrates. See, carbohydrates are divided into 3 classes. We have the monosaccharide, we have the oligosaccharide, and we have the polysaccharide. So we are going to start with what? Monosaccharides. So monosaccharides are carbohydrates which are rather called simple sugar molecules. They are simple sugar molecules because their molecule is this one. Example of such is glucose. So, and glucose is just, you are familiar with glucose used in sports by footballers, so in order to supply energy. So those are the basic example of monosaccharides. So they are molecules. They are like in discussion of polymers. They are always a molecule when joined together or linked together. They produce a oligosaccharide or polysaccharide. If you join much more larger numbers of monosaccharides together, you form polysaccharide. Now, oligosaccharide, that is just the definition for monosaccharide. We don't have much to discuss. when we get to oligosaccharide, we discuss few about what disaccharide, which is just a member of oligosaccharide. So in oligosaccharide, we have the what? Monosaccharide, we have the disaccharide, we also have the trisaccharide, we have the tetrasaccharide and pentasaccharide, and so on. So let's look at how there is linkage between monosaccharide to form disaccharide or trisaccharide. When you say oligosaccharides, it's just combination of disaccharides, trisaccharides up to pentasaccharides, meaning the joining together of what? Two monosaccharides form disaccharides, joining together of three monosaccharides forms trisaccharides, and joining together of four monosaccharides forms tetrasaccharides. So let's look at the definition for disaccharides. So we can, since there are two molecules of monosaccharide linked together to form the disaccharide, so we can define it as a molecule or a carbohydrate which, when on hydrolysis, produces two molecules of monosaccharide of simple sugar. So when you hydrolyze, I mean when you perform hydrolysis, or an hydrolysis reaction of disaccharide produces what? Two molecules of monosaccharide. When oligosaccharides is a general term to qualify disaccharide, trisaccharide, pentasaccharide, tetrasaccharide. So when you perform hydrolysis for oligosaccharides, you break down into what? Between 2 to 10 molecules of simple sugars, which are the monosaccharides. When you perform hydrolysis of oligosaccharides, which are these members, they are just between 2 to what? To 10. So when you break them down on hydrolysis, they produce what? Between 2 to 10 molecules of simple sugars. So let's look at some disaccharides, which when you hydrolyze, they produce what? 2 molecules of monosaccharide. Example of such is, we have maltose, lactose, we have cellulose. So these are disaccharides. So we are just going to look at how they are linked at the elementary level, since this is an introductory course. So we are going to look at how they are linked. So they call it glycosidic, because when you link two glucose of the monosaccharide, I told you an example of monosaccharide is glucose. When you link two glucose of the monosaccharide together, there will be a linkage. So they call it glycosidic linkage. So let's look at the glycosidic linkage in maltose. So though the glucose in the initial form of which I have drawn earlier will not be on a straight chain, it's going to be in a cyclic chain. So let's draw that and see. We have so these angles here represent the position of a carbon. So this is an example of glycosidic linkage of two glucose. Here you have hydrogen, and you have, this is first carbon, the second, the third, the fourth, the fifth, and the sixth is CH2OH. So then we have OH here, we have H here, then OH here, OH here, then H here, H here, H here, then we have OH, then H. Here we also have OH, OH, then H, then we have 6H2OH, Then we have H with OH. So this is maltose molecule. This is molecule of the maltose. One more point to mention: the carbon here, the carbon at this one. So the linkage, before I made mention of this carbon, the linkage takes place between the carbon. This is the carbon 1 here, 2, 3, 4, 5, 6. When you number, and this is carbon 1, 2, 3, 4, 5, 6. So the linkage is between carbon 1 and 4. So that's where the linkage exists. That's where the glycosidic linkage exists in maltose. Now, emphasizing on this carbon here. This carbon is an alpha anomer. It is an anomer, just like the case of when I was mentioning chiral. Chiral means when you are attached to molecules of atoms of different species. So this carbon is attached to 4 different species of atom. It's attached to hydrogen here, attached to OH here, attached to carbon here because it's directly attached to this carbon here, and attached to oxygen here. So that's 4 different species of molecule. So that is why it's called anomer. And it is called an alpha anomer because the OH here is pointing downward. So it is called alpha anomer. It's at carbon C1. So carbon C1 here, I'm talking about, is a what? It's an alpha anomer, and this is the molecule of maltose. So on the aspect of cellulose, the cellulose is the same molecule as maltose, just like cellulose has a beta anomer. The beta anomer is also at this point telling us that what? The OH has turned the other way around, upside down, and the difference in the linkage is just here. I will draw that of cellulose as well. So let's have the structure of this nature. So we have OH, H, this is a cellulose now. CH2 due to availability of space. And we have O here. H, OH, H. H is here, and we have CH2OH. So this is what we call the cellulose. And the carbon here is also an anomer, but it is called a beta anomer. Because it's a beta anomer because this OH group is pointing upward. So the same molecule of the cellulose is equivalent to that of similar to that of the structure is similar to that of lactose, but just that lactose also have alpha anomer, and the linkage here is also at 1 and 4, and the similar type of linkage present in cellulose is what is present in lactose. So these are just preliminary structure of disaccharides and how they are formed. Then for polysaccharides, these consist of large molecules of monosaccharides. So we are going to continue from there in the next video. Thanks for watching our YouTube video. Please subscribe to our YouTube channel. We are going to bring you more videos. Thank you.